Signaling in single-celled organisms. Within-cell signaling allows bacteria to respond to environmental cues, such as nutrient levels. Some single-celled organisms also release molecules to signal to each other. Signaling in yeast. Yeasts are eukaryotes, fungi, and the components and processes found in yeast signals are similar to those of cell surface. Receptor signals in multicellular organisms. Budding yeasts, Figure 1, are able to participate in a process that is similar to sexual reproduction that entails two haploid cells, cells with 1/2 the normal number of chromosomes, combining to form a diploid cell, a cell with two sets of each chromosome, which is what normal body cells contain, in order to find another haploid yeast cell that is prepared to mate. Budding yeasts secrete a signaling molecule called mating factor. When mating factor binds to cell surface receptors in other yeast cells that are nearby, they stop their normal growth cycles and initiate a cell signaling cascade that includes protein, kinases and GTP binding proteins that are similar to G proteins. Signaling in bacteria. Signaling in bacteria enables bacteria to monitor extracellular conditions, ensure that there are sufficient amounts of nutrients, and ensure that hazardous situations are avoided. There are circumstances, however, when bacteria communicate with each other. The first evidence of bacterial communication was observed in a bacterium that has a symbiotic relationship with Hawaiian bobtail squid. When the population density of the bacteria reaches a certain level, specific gene expression is initiated, and the bacteria produce bioluminescent proteins that emit light. Because the number of cells present in the environment, cell density, is the determining factor for signaling. Bacterial signaling was named quorum sensing. In politics and business, a quorum is the minimum number of members required to be present to vote on an issue. Quorum sensing uses autoinducers as signaling molecules. Autoinducers are signaling molecules secreted by bacteria to communicate with other bacteria of the same kind. The secreted autoinducers can be small, hydrophobic molecules such as acyl homoserine lactone, AL, or larger peptide-based molecules. Each type of molecule has a different mode of action. When AL enters target bacteria, it binds to transcription factors, which then switch gene expression on or off. Figure 2. The peptide autoinducers stimulate more complicated signaling pathways that include bacterial kinases. The changes in bacteria following exposure to autoinducers can be quite extensive. The pathogenic bacterium, Pseudomonas aeruginosa, has 616 different genes that respond to autoinducers. Some species of bacteria that use quorum sensing form biofilms, complex colonies of bacteria, often containing several species, that exchange chemical signals to coordinate the release of toxins that will attack the host. Bacterial biofilms, Figure 3, can sometimes be found on medical equipment. When biofilms invade implants such as hip or knee replacements or heart pacemakers, they can cause life-threatening infections. Research on the details of quorum sensing has led to advances in growing bacteria for industrial purposes. Recent discoveries suggest that it may be possible to exploit bacterial signaling pathways to control bacterial growth. This process could replace or supplement antibiotics that are no longer effective in certain situations. Evolution connection. Cellular communication in yeasts. The first life on our planet consisted of single-celled prokaryotic organisms that had limited interaction with each other. While some external signaling occurs between different species of single-celled organisms, the majority of signaling within bacteria and yeasts concerns only other members of the same species. The evolution of cellular communication is an absolute necessity for the development of multicellular organisms. and this innovation is thought to have required approximately 2.5 billion years to appear in early life forms. Yeasts are single-celled eukaryotes, and therefore have a nucleus and organelles characteristic of more complex life forms. Comparisons of the genomes of yeasts, nematode worms, fruit flies, and humans illustrate the evolution of increasingly complex signaling systems that allow for the efficient inner workings that keep humans and other complex life forms functioning correctly. Kinases are a major component of cellular communication, and studies of these enzymes illustrate the evolutionary connectivity of different species. Yeasts have 130 types of kinases. More complex organisms such as nematode worms and fruit flies have 454 and 239 kinases, respectively. Of the 130 kinase types in yeast, 97 belong to the 55 subfamilies of kinases that are found in other eukaryotic organisms. The only obvious deficiency seen in yeasts is the complete absence of tyrosine kinases. It is hypothesized that phosphorylation of tyrosine residues is needed to control the more sophisticated functions of development, differentiation, and cellular communication used in multicellular organisms. Because yeasts contain many of the same classes of signaling proteins as humans, these organisms are ideal for studying signaling cascades. Yeasts multiply quickly and are much simpler organisms than humans or other multicellular animals. Therefore, the signaling cascades are also simpler and easier to study, although they contain similar counterparts to human signaling. Section summary. Yeasts and multicellular organisms have similar signaling mechanisms. Yeasts use cell surface receptors and signaling cascades to communicate information on mating with other yeast cells. The signaling molecule secreted by yeasts is called mating factor. Bacterial signaling is called quorum sensing. Bacteria secrete signaling molecules called autoinducers that are either small, hydrophobic molecules or peptide-based signals. The hydrophobic autoinducers, such as all, bind transcription factors and directly affect gene expression. The peptide-based molecules bind kinases and initiate signaling cascades in the cells. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.